and today we're covering the cardiovascular system blood. So let's go ahead and begin. Oops, sorry. Blood composition. Blood is a fluid connective tissue composed of plasma and formed elements. Formed elements include erythrocytes, red blood cells or RBCs, leukocytes, white blood cells or WBCs, and platelets. Now we used to call them cells, but now we don't use cells, we use formed elements because only leukocytes are true cells. Erythrocytes, they lack nucleus and major organelles, and platelets are fragments of cells. That's why we change definition of blood as a connective tissue composed of plasma and formed elements. Hematocrit is a percent of blood volume that is red blood cells. Now, if we draw blood and put it in a test tube and then centrifuge the tube, then components of blood will be divided based on how heavy they are. So on the bottom, we will have heaviest material, and those going to be erythrocytes. Then Buffy code, less than 1% of whole blood, is that include leukocytes and platelets. And on the top of test tube, you will find plasma. That is about 45% of whole blood. It's the least dense component. Now, normal hematocrit is about 45%, a little bit higher for males than females. 47 plus minus 5% for males and 42 plus minus 5 for females. Physical characteristic of blood: sticky, opaque fluid, color scarlet to dark red, pH 7.35, 7.45, temperature 38 degrees Celsius, about 8% of body weight, and average volume is 5 liters. So a little bit more for males, 5 to 6 liters, and less for females, 4 to 5 liters. Functions of blood include distribution. Distribution of what? Oxygen and nutrients to body cells, metabolic waste to the lungs and kidneys for elimination, and hormones from endocrine organs to target organs. Regulation is another function. Your blood regulates body temperature by absorbing and distributing heat, normal pH using buffers, and adequate fluid volume in the circulatory system. And another function of blood is protection against blood loss. Plasma proteins and platelets initiate clot formation and protection against infections. You have antibodies, complement proteins, white blood cells that defend against foreign invaders. Blood plasma is about 90% water. The rest are proteins and other chemicals. Proteins are mostly produced by liver. Out of these proteins, 60% albumin, 36% globulins, and about 4% fibrinogen. In addition, blood plasma consists of nitrogen byproducts of metabolism. So you will find, it's not consist, it's you will find in blood plasma, in addition to water and proteins, also nitrogenous byproducts such as lactic acid, urea, creatinine, nutrients, glucose, carbohydrates, amino acid, electrolytes, sodium, potassium, ions, and so on, respiratory gases, oxygen, carbon dioxide, and hormones. Formed elements. Only white blood cells are complete cells. RBCs have no nuclei or organelles, major organelles. They do have some, but they don't have mitochondria, for example, no nucleus. Platelets are cell fragments. Most foreign elements survive in the bloodstream for only a few days. Most blood cells originate in bone marrow and do not divide. Erythrocytes, but by concave disc and nucleate, that means no nucleus, essentially no organelles, filled with hemoglobin for gas transport as a major factor contributing to blood viscosity. About 5 million red blood cells per microliter of blood. Structural characteristics contribute to gas transport. It's biconcave shape. And if you go back to previous slide, you can see what biconcave shape is. So it's not just a flat, I mean, it is kind of flattened in the middle. Right, so that's, you see this biconcave shape on the both sides. Okay, I'm sorry. Biconcave shape, huge surface area relative to volume. More than 97% is hemoglobin, not counting water. No mitochondria. ATP production is anaerobic. No use of oxygen in generation of ATP. A superb example of complementarity of structure and function. RBCs are dedicated to respiratory gas transport. Hemoglobin binds reversibly with oxygen. Each red cell contains about 280 million molecules of hemoglobin. Hemoglobin structure, it's made of protein globin, 2 alpha and 2 beta chains, heme pigment that bounded to each globin chain. Atom of iron in each heme can bind to 1 oxygen molecule. Each hemoglobin molecule can transport 4 oxygen molecules. So here is the structure. Of course, it's just a picture, but you can see that that's a molecule of hemoglobin. And what's shown here, brown and tan color, those are protein chains, right? So we have, look, one protein chain or polypeptide, would say, one polypeptide, another polypeptide, it's a beta globin chain, and lighter color, alpha globin chain. So those are polypeptide. That means proteins made of amino acids. Each polypeptide has a pigment, heme. Inside the heme, shown here in kind of green color, that's iron. So you can see over here, iron in the middle, and that's a structure of heme. Now, each atom of iron can bind 1 molecule of oxygen. That means 1 molecule of hemoglobin can carry 1, 2, 3, 4 molecules of oxygen. Now, hemoglobin reversibly binds to oxygen. Now, where does it happen? Well, where would you have oxygen? In your lungs, right? So we have oxygen loading in the lungs. And when this happens, we produce oxyhemoglobin, that is ruby red. Now, where you unload oxygen, you unload in the tissues, because your whole goal is to deliver oxygen from lungs to the tissue. So you load oxygen in the lung, unload in the tissue. Now, when that happens, you produce deoxyhemoglobin. or reduced hemoglobin that is dark red. You also gonna load carbon dioxide in the tissue. Why? Because you deliver it to the lungs. Then we produce carbamina hemoglobin. Actually, most of the oxygen is carried inside the plasma of red blood cells. Oh, I'm sorry, inside the cytoplasm of red blood cells. Some carried by plasma of blood. Only 20% of carbon dioxide will be carried in the form of carbamina hemoglobin. Hematopoiesis is blood cell formation. Occurs in the red bone marrow of axial skeleton, girdles, and proximal epiphysis of humor and femur. Fate and distribution of erythrocytes. A lifespan of erythrocytes is 100 to 120 days, so about 1 month. Old RBCs become fragile and hemoglobin begins to degenerate. Macrophages engulf dying RBCs in the spleen. Heme and globin are separated. Iron is salvaged for reuse. Heme is degraded to yellow pigment, bilirubin. Liver secretes bilirubin in bile into the intestine. Degraded pigment leaves the body in feces as stercobilin, and globin is metabolized into amino acids. Okay, so I just read you this description. Now let's explain it a little bit in more detail. Okay, so let's start with your kidneys. Now, when blood flows through the kidney, if you have low oxygen level in your blood, that low oxygen level in blood stimulate kidney to produce erythropoietin. That's a hormone. And erythropoietin leaves kidney and enter your bloodstream. So the level of erythropoietin in blood rises. Erythropoietin and necessary raw material in blood promote erythropoiesis in red bone marrow. So now when you have high level of erythropoietin, it promotes synthesis of red blood cells. Where? In a red bone marrow inside your bones. Now new erythrocytes enter bloodstream and function about 4 months, right? So that makes sense. Your kidney sends all oxygen level low. Let's tell your body that you need more red blood cells. How to tell it? We have a special messenger, erythropoietin. So we're going to secrete erythropoietin and it will stimulate formation of new erythrocytes. For 120 days, they just inside your circulation and carry oxygen, right? So do their job. What happened after that? Aged and damaged red blood cells are engulfed by macrophages of liver. So your blood flows through the liver, right? Through the spleen, through the bone marrow. Well, it flow. You do have capillaries in the bone marrow, capillaries in spleen, capillaries in the liver. So when blood carries through, flows through those organs, the old and damaged, aged and damaged red blood cells are destroyed by macrophages. Right. So here you have your cell, red blood cells with a lot of hemoglobin inside. Your macrophage is gonna eat it up, break it down. Now, what happened with hemoglobin? Hemoglobin will split into heme and globin. Remember, globin is protein. Protein is made of amino acid, so you break it down into amino acid, amino acid back to your bloodstream, and you reuse it, right? You like when you eat steak, you use this amino acid to build your own proteins. Here, you break down globin into amino acid and you recycle it. Now, what happened with heme? Well, heme is degraded to pigment, bilirubin. Remember, heme is made of pigment and iron. So this heme that degraded in bilirubin, it enters your kidney or liver. So liver use bilirubin to make bile, right? So bilirubin is picked up from blood by liver, secreted into intestine in bile, metabolized to stercobilin by bacteria, and excreted in feces. Next, well, and excreted in feces, I would say, go this way. Right, so again, so hemoglobin degraded into heme, that is a pigment. Heme as a pigment became bilirubin. Bilirubin picked up by liver. Liver make bile. Bile secreted into small intestine. Then it's metabolized by bacteria and secreted with the feces. Now, iron from a heme stored as ferritin and hemosiderin, so those are chemicals that stores this iron. Then iron is bound to transferrin and released to blood from liver as needed for erythropoiesis. Right, so the iron stored as ferritin. Ferritin is stored in liver, and when you need it, ferritin get attached to a special carrier called transferrin and released to the blood, right? So the blood can bring it to bone marrow and synthesize more erythrocytes, right? And plus, of course, food nutrients, including amino acid, iron, B12 vitamin, folic acid, are absorbed from intestine and enter the blood. So if you now in your blood, if you have iron, you have vitamin B12, folic acid, amino acids, right? Your blood bring it to a bone marrow. Also, your blood brings erythropoietin, that is hormone, to a bone marrow, and you can synthesize more new erythrocytes, right? So fate and distribution of erythrocytes includes synthesis, formation inside bone marrow, and then recycling in liver or spleen by macrophages, storage of necessary in material like iron, and then release it back to a bloodstream when you need for new synthesis of erythrocytes. Erythrocyte disorders, anemia. Anemia defined as blood that has abnormally low oxygen carrying capacity. We have about 400 types of anemia. It's a sign rather than disease itself. Blood oxygen levels cannot support normal metabolism. It's accompanied by fatigue, paleness, shortness of breath, and chills. We have different types of anemia. You already see over here 400 types. The most common is iron deficiency anemia, vitamin deficiency anemia, especially vitamin B12 and folate. And I think that's it for erythrocytes. I thought I have another slide, but that's about all the information that we're gonna cover today about erythrocytes. Now, what other form element we have in our blood? Leukocytes. make up less than 1% of total blood volume, can leave capillaries by diaperesis. You don't have many cells in your body that can move on its own. You actually have only 2 cells that are capable of movement. Sperm, they have flagellum, and it allows sperm to move, and white blood cells. They move by diaperesis, like amoeba. Leukocytes move through tissue spaces by amoeboid motion and positive chemotaxis. Positive chemotaxis means you have a chemical that attract those white blood cells. The white blood cells count is over 11,000 per cubic millimeter, and it's a normal response to bacterial or viral infection. We have 2 types of leukocytes, granulocytes and agranulocytes. I think here you need a micro, or maybe because it's cubic. You know, this I need to check. Let me check it right now. Let me pause this video for a second, and I will tell you for sure if it's correct number or not. Okay, so I'm back. So I change it because in the previous slide we have red blood cells count is 5 millions per microliter. So I change this cubic millimeter to microliter as well, so we can see the difference, right? I guess cubic milliliter is equals microliter. I'm not sure about it. I'm not that good with all these dimensions. But white blood cells, we have about 10,000 per microliter. When it's over 11,000, that's response to infection. So let me show you where we have this red blood cells count. So erythrocytes, we have 5 million red blood cells per microliter of blood. So if you take 1 microliter, you will find 5 million of red blood cells, and you will find only 10,000 of white blood cells, right? So now it makes more sense. So when we have count over 11,000 per microliter, then it's normal response to bacteria or viral invasion. That means some infection going on. Now leukocytes are divided into 2 groups, granulocytes and agranulocytes. Now, you probably heard about differential white blood cells count. So his normal amount is from 5 to 10,000 in 1 microliter. And if you look at the white blood cells, then you will notice over here that the most numerous type of white blood cells are neutrophils. And then we have lymphocytes. After that, we have monocytes, eosinophils, and basophils. We don't need to memorize those numbers, but we need to remember relative concentration of those cells. There is mnemonic to remember, and mnemonic, let me draw it. Mnemonic says never let, so hold on, it disappears. I don't want it to disappear. Never let monkeys eat bananas. So you can remember that. And for neutrophils, so the most numerous neutrophil, lymphocytes, monocytes, eosinophils, and basophils. Right. You also can see that neutrophils, eosinophils, and basophils, they are granulocytes, and lymphocytes and monocytes, agranulocytes. So granulocytes are neutrophils, eosinophils, and basophils. Cytoplasmic granules stain specifically with right stain. So why we call them granulocytes? Because if we use special technique to stain those cells, so we can see them under microscope, we will see lots of granules. That's why we call them granulocytes. They larger in size and shorter lived than RBCs. They have lobe nuclei, and all of them are phagocytic. So here's neutrophils. If you look at this histology slide, this is red blood cell and white blood cell. Here, neutrophil is a bigger cell. This dark purple stuff over here, I don't know why. Hold on a second. Kind of, I don't know why I'm drawing something, right? And at the same time, it's just it's not doing the. Let me see. Okay, here now it's better. So this is neutrophil. You can see granules, right? So it's a granulocyte. This purple structure here, that's a nucleus. And nucleus has several lobes. That's why we say it's a nucleus is made of several lobes, lobed nuclei. So neutrophils are most numerous WBCs, polymorphonuclear leukocytes, or sometimes they call them polis, or PMNs. Fine granules take up both acidic and basic dyes, give the cytoplasm a lilac color. Granules contains hydrolytic enzymes or defensins and very phagocytic bacteria slayers. Eosinophils, red staining bilobed nuclei. So here's our eosinophil. You see lots of granules again. Now look at this nucleus. It's made of 2 lobes right there. That's why it's bilobed nuclei. Red to crimson, acidophilic, has a lysosome-like granules, digest parasitic worms that are too large to be phagocytized, modulator of the immune response. So let's go kind of back, because neutrophils are the major white blood cells that fight bacteria. So if you have elevated number of neutrophils, you have lots of neutrophils, that can be possibly because of the bacterial infection. If you have lots of eosinophils in the patient blood, that either patient have some parasitic worms, or we have elevated level of eosinophils when patient have allergy. Basophils, rarest white blood cells, large purplish black basophilic granules containing histamine. Histamine is inflammatory chemical that acts as a vasodilator and attracts other white blood cells to inflamed sites. Vasodilator means it dilates your blood vessels. They functionally similar to mast cells. So here's a basophil with a lot of granules, and it's a dark purplish black color. Now, if you see this name basophil, because it pick up basic dye, and eosinophil, it picks up acidic dye, so that's became a red. And neutrophils, they pick up both acidic and basic dyes. So we use special technique to make those cells visible. Most of the cells in your body are translucent. Like red blood cells, yes, they do have color because of hemoglobin, but white blood cells, you will not be able to see those cells. You need to dye them. So we use a special technique to dye those cells. And a technique includes using both acidic and basic dyes. So some cells, their granules pick up both acidic and basic dye. And acidic is red and basic, is blue. So when you mix red and blue, that give you lilac color for neutrophils. If you have blood cells that pick up only acidic dye, then they're gonna be red. And if you have the ones that pick up only basic dye, that's gonna be almost dark blue or like purple. That's why that's where names came from: basophils, eosinophils, neutrophils, and name granulocytes we use because you see those visible granules. Okay, so here's our granulocytes. You can compare how they look using the same dye, the same technique. You use the same dye, but they look differently. So here's a neutrophil, eosinophil, and basophil. Now, granulocytes include lymphocytes and monocytes. They lack visible cytoplasmic granules, have spherical or kidney-shaped nuclei. Lymphocytes, large, dark purple circular nuclei with a thin rim of blue cytoplasm. So if you look over here, the lymphocytes is the smallest white blood cell. So when I see this L, that remind me it's very little. So it's a little bit bigger than red blood cell, but look at this huge round nucleus. So those small cells with very important function. Mostly you find them in lymphoid tissue. Few circulate in the blood, and they're critical to immunity. Why? Because they can we have two types of them. T cells that act against virus-infected cells and tumor cells, and B cells. And B cells can differentiate into plasma cells and produce antibodies, right? So here's our B and T cells, and those are actually lymphocytes. And another granulocyte is monocyte. And guess what? It starts with M, and it's remind me about big monster. This is the biggest white blood cell. So look at this, huge. It's the largest leukocytes, abundant pale blue cytoplasm, so that's a cytoplasm, dark purple staining U or kidney-shaped nucleus. They leave circulation, enter tissues, and differentiate into macrophages, actively phagocytic cells, critical against viruses, intracellular bacterial parasites, and chronic infections. Activate lymphocytes to mount an immune response. So they activate lymphocytes, your monocytes. Right, so let's see if we have, okay, so here's our small lymphocytes and huge monocyte. Right, disorders of leukocytes. You have leukopenia, too few leukocytes, leukemia, abundance of leukocytes, cancer, lymphoma, malignant cancer that affect T and B or both T and B lymphocytes. Next, platelets, small fragments of megakaryocytes, blue stained outer region and purple granules, form a temporary platelet plug that helps seal breaks in blood vessels. So here you can see megakaryoblast. So first, we're going to start with a stem cell. hematocytoblast, that's a stem cell that will differentiate into megakaryoblast. Then a promegakaryocyte, then we have this megakaryocyte, and you see those little fragments that breaks off of this huge cell, they form platelets. And platelets is important in hemostasis or stopping bleeding, because they form a platelet plug that seals this breakage in blood vessels. Platelet disorders, thrombocytosis, when you have too many platelets. If you look at this blood smear, you see lots of these little dots, large amount. That's too many platelets. Thrombocytosis. And thrombocytopenia, too few platelets. Now go ahead and look at this blood smear, and I see like one here, maybe one over there, maybe one here, and that's about it. So if you compare two slides, you will see too many or too few platelets, and neither one is good for your health. And this is the last slide. Okay, so thank you so much for watching. Just want to remind you that we cover cardiovascular system blood, and I hope it was helpful.